Hello and welcome to the ROSA Worldwide webinar series. My name is Mike Reed. I'm the Chief Scientist at ROSA, and our speaker today is Melissa Hallow. Melissa is an Associate Professor at the University of Georgia with joint appointments in the College of Engineering and the College of Public Health. She earned her PhD from the Georgia Institute of Technology and worked for several years at Novartis Pharmaceuticals before making the move to academia at the University of Georgia. Her research group uses mathematical models of renal and cardiac physiology and pharmacology to better understand disease processes and drug mechanisms in hypertension, kidney disease, heart failure, and drug-induced kidney injury. Just a brief note that we'll handle questions at the end of the talk today, and you can ask questions by typing them into the questions window on your control panel to the right of your screen. Okay, so Melissa is the presenter. So with that, welcome, Melissa, and we're looking forward to your talk.
Yep, thanks, Mike, and thanks for this opportunity to present. I'm going to turn my camera off now so that it doesn't interfere. But yeah, so the talk I'm going to give today, if any of you guys attended my talk at ACOP, I apologize because it's very much a repeat of that talk, but it's, I think I like to give this talk because it's a really nice story, I think, of how multi-scale modeling can be applied over the course of the process of drug development in many different aspects of the development of a compound, and how it can make impact, and it is kind of a complete story, I think, anyway. So I hope you guys enjoy it. Before I get started, I just want to acknowledge, in case I run out of time at the end, all the many people who have been involved in this work, both contributing directly to it and many of our collaborators and also our funders as well. So first, maybe just starting from big picture, some of the major challenges across drug development currently. One of the big challenges is, just understanding mechanisms of disease when we're talking about complex multi-organ systems, right? So when we're talking about kidney disease, it's not just the kidney, right? It's the heart and it's the vasculature. Same thing with heart failure, right? All of these organs interact, and the biomarker responses that we observe are often.
Excuse me, Melissa, are you advancing your slides?
I am. Hold on a second. Oh, here we go. I know the problem. Slide. There we go. I've only gone. I'm sorry. Yeah. Let me just go back one slide and make sure that I acknowledge all of my collaborators and lab members. It's all you've missed so far. But yeah, so when we're talking about these multi-organ diseases, often the biomarkers that we observe can be unintuitive and difficult to understand, and that makes it difficult to make decisions when we don't really know what those biomarker changes mean. Often, too, there are challenges in understanding and predicting drug safety signals. So sometimes when we see an early signal, it's difficult to interpret what that means long-term for the safety of a compound. In addition, particularly in cardiorenal disease, the pivotal trials, the phase 3 trials, are extremely expensive because they have to be large and they have to be pretty long trials. And so de-risking those trials is super important, right? And making sure that we choose the right population and that we really understand how our drug is working, and so that we can, really support the likelihood that this drug is going to be successful when we start looking at outcomes. And then lastly, for almost any disease in the cardiorenal space, we're always talking about combination therapy. So understanding how our drug works when we add it on to standard of care, or when additional new drugs come on the market, how those drugs are going to interact with our drug of interest is super important. So before I go into how we address those questions, I first just want to talk about the cardiorenal system in general, right? So in the cardiorenal system, normally we want to maintain homeostasis, and what we mean by that is that the body wants to maintain steady blood flow, so this constant rates of delivery of blood, which includes our oxygen and nutrients, and clearance of waste products to each of our organs. Our body wants to do that at stable concentration, so it's super important that we maintain maintain normal sodium concentrations and potassium and chloride and calcium and protein concentrations. All of these things impact the ability of our body to function normally, right? So just simple, fundamental processes like the generation of action potentials in our heart and in our neurons depends on having a stable, fixed concentration of electrolytes. And the cardiorenal system needs to maintain the stable blood flow and stable concentrations, ideally at stable pressures. So generally we have higher pressures on the arterial side, and those pressures normally fall to near zero as you approach the return to the heart. So if we think about these three things, flow rate, concentrations, and pressure, right? So flow rate is just a function of the pressure drop and the resistance of the vasculature. Concentration is just the ratio of the amount of a substance to its volume. And then pressure is proportional to the volume of fluid divided by the capacitance, or the space available for that fluid. So if we think about those three things together, right, there are a limited number of things that the cardiorenal system is able to control in order to maintain homeostasis, right? So there's amount, volume, pressure, resistance, capacitance. So a big tool that the cardiorenal system uses is changes in vascular resistance, so constricting and relaxing of our arteries, and also a capacitance, which is more vasodilation of our venous system. In addition, there are the other thing that the cardiorenal system can control is both the volume of fluid in our body and the amount of electrolytes. And that's really the job of the kidney. So the job of the kidney is to match the amount of sodium and water and other electrolytes coming in to the amount that's leaving. Okay. And so anytime there is a sodium imbalance in the body, it's always going to be corrected over time. So what the kidney does, kind of above all else at the cost of everything else, is that it's going to return sodium balance. And what we mean by that is that just that your excretion equals intake. If you don't have sodium balance, if you're taking in more sodium than you're excreting, then you're going to retain water, and the volume of fluid in the body is going to grow. You're going to retain water because the body wants to keep that constant sodium concentration, right? And if you're taking in less sodium than you're excreting, then you're going to lose water. So in a normal situation, when there's an increase in sodium intake, the kidney quickly adapts to increase the amount of sodium and water that's being excreted. And so there's a fast correction, and there's a pretty small change in the amount of sodium and water that's retained. And this results in potentially a small change in blood pressure. Actually, in some situations, you can even have no change or blood pressure going down because the system adapts in other ways. But what can happen in disease states is when this adaptation or when this adjustment occurs more slowly, so you have a much slower correction process, it leads to a larger accumulation of fluid, more volume, right? And therefore, if you have more volume, the same capacitance, you end up with higher pressures. So how does the kidney know how to do this, right? Like the kidney doesn't really have a brain, it's not thinking, right? How does it know how to correct, and what determines the speed at which it does that, right? That turns out to be a really complicated question, but it's a question that that's really important to understand and to be able to model how the body is able to control these processes. So if we think a little bit about kidney physiology, right? If we think about from the organ to the organ system to the organ, which is a kidney, the working unit of the kidney is the nephron, which is comprised of a glomerulus and a tubule. If we look at this glomerulus, there is this organ substructure, right? We have the glomerular capillaries and then the tubule here. If we go in and look further at this, this kind of a nice, pretty picture of the glomerular capillaries, right? The filtration rate, which is this key measure of kidney function, the glomerular filtration rate, is really a function of how many of these nephrons you have, how permeable this membrane is, and the net filtration pressure across this membrane. Okay? But then once we filter fluid across this glomerular membrane, that filtrate flows along the tubule, so along this complex tubule system, and there are all these various processes involved in reabsorbing most of what you filter. So 99% of what gets filtered gets reabsorbed as you move along the tubule at different rates in different segments of this nephron tubule. And if we look even closer, kind of thinking about the multi-scale nature of the system, right? Well, how does it know what to reabsorb? Well, and what to filter, right? Well, these are just tissues, right? So the glomerulus is just made up of a bundle. It's just this bundle of capillaries that's made up of endothelial cells and mesangial cells and podocytes. And then the tubules are just these tubes of epithelial cells. And if we look a little closer at those epithelial cells, right, within a cell, there are transporters that lie on the basal and apical surface of the cell that control the amount of flux of ions and water across the cell. And then if we look at the molecular level, even we can think about this from even that tiny scale, right, of, for instance, hormones like aldosterone that diffuse into the cell and bind to receptors inside the cell and affect the behavior. Or if we look at the transporter that I'm going to talk a lot about later, sodium glucose cotransporter 2, that's also another transporter that's on the cell of these membranes. So it's really this multi-scale system, right? And it doesn't have like one unique control mechanism, right? Instead, it's a kind of an integrated response to changes. So if we come back to that question of sodium balance, right? Sodium balance means that intake equals excretion. And excretion is just the difference between what you filter and what you reabsorb. So if there's a sodium imbalance, you can really correct that only in two ways. You can either change what you're reabsorbing, and that's what a normal kidney normally does, and GFR normally stays constant, or you can change how much you're filtering. So if the kidney's not able to adapt by changing what you reabsorb, then it's going to change what's filtered. But ultimately, it's going to get back to sodium balance. So how does it do these things? How does the kidney change these things? Well, there is a system of both intrinsic and biophysical and neurohormonal processes and feedbacks that can lead to alterations in tubular reabsorption rates, so the rates at which things are getting reabsorbed along this nephron, or changes in renal vascular resistances that can result in changes in how much ultimately gets filtered. And if we look at that, it's really, I won't go into great detail here, but we've spent a lot of our work in developing models of the system has been in trying to understand all the various feedbacks that are involved in the system. So there's the renin-angiotensin-aldosterone system. There are processes such as sensing of sodium flow through the macula densa. So if sodium delivery to this particular portion of the nephron changes, there is a special set of cells in that region called in the macula densa. These cells stimulate renin secretion, and they also stimulate afferent vasodilation or vasoconstriction. So that's kind of one example of the way that the kidney can control particular biophysical signals, right? So sodium delivery, blood pressure, central venous pressure, renal interstitial hydrostatic pressure, sodium concentration, cardiac output. All of these are biophysical signals that are sensed and responded, and there are neurohormonal mechanisms and some intrinsic vasoactive mechanisms as well that lead to changes in things like resistance, reabsorption rates, that ultimately then can return homeostasis. Okay. So all of those are the processes that happen in a normal kidney, right? We're able to sense these different changes in biophysical signals, apply the appropriate neurohormonal response to return homeostasis. But what happens when that homeostasis is disrupted? So when the body can no longer maintain constant flow rates or concentrations, for instance, if the heart is injured, or there's kidney injury, so it can't excrete as much as it normally does, or there's some vascular or neurohormonal changes, if something causes either flow rates or concentrations to be disrupted, the system is going to adapt to that, and it's going to adapt to restore flow rate because it's necessary for life that our organs receive the proper amount of blood, and it's going to restore concentrations, and it usually does this at the expense of pressure. So in most disruptions of cardiorenal homeostasis, what happens is pressure goes up in all or some component of the system in order to restore sodium concentration, restore flow rates. And this can lead to hypertension. It can lead to increased filling pressures on your heart and subsequently long-term to heart failure. It can lead to elevations in pressure in the glomerulus, which is one of the big drivers of kidney disease. And so all of these elevated pressures start causing damage over time, right? Cause fibrosis, inflammation, organ damage, and ultimately morbidity or mortality over time, right? So these corrections or these adaptations may work for a long time until that compensatory capacity is exceeded. And generally, when we're trying to treat cardiorenal disease, one of the big challenges is we often don't know what the underlying cause of an individual's disease is, or even if we know what it is, often it's difficult to address the underlying cause, right? So the therapies that we're developing are often trying to move that homeostasis set point to a more tolerable state, right? To lower those pressures in some way so that we can maintain flow rate or concentrations at a lower pressure. So it's almost like we're trying to trick the kidney into doing its job at a different pressure point. And another one of the big challenges of this is that often when we're trying to develop therapies that do this, we often get kind of unintuitive or conflicting biomarkers because our responses, because we're often not able to measure the thing we really want to measure. And we're often also talking about processes that happen over a range of timescales, right? So there are neurohormonal responses that happen really quickly that can lead to changes that happen over long periods of time. And one of the biggest challenges is, for instance, most of the drugs that we give to treat kidney disease, they cause sodium, an increase in sodium excretion, but it's temporary. You only see it, in the first few days, and then that sodium excretion response goes away. You can't measure it anymore. But the system has adapted to being able to excrete a normal amount of sodium at a lower pressure. Okay, so that's kind of a big picture. What are some of the challenges in cardiorenal drug development? I'm going to talk now more specifically about the case of SGLT2 inhibitors. Right, so when SGLT2 inhibitors were first approved, they were approved to treat type 2 diabetes, and they seem kind of like a Me Too drug. They lowered HbA1c about the same as every other anti-diabetic drug at the time. And the mechanism seemed straightforward, which I'll talk about in just a second. But even though it seemed to have this straightforward mechanism, it was noted that it consistently caused an initial decrease in GFR, and that was really concerning because there was no understanding of why this drug was causing GFR to drop and what that meant, whether it was doing something unknown to the kidney. So a lot of concern about that. So the mechanism for SGLT2 inhibitors basically is in the proximal tubule of the kidney. So this is a kidney, this is a nephron, right? The upper section of this tubule is the proximal tubule. And normally what happens is you have glucose in your bloodstream, you filter glucose across the glomerulus, and in a normal person, all of that glucose gets reabsorbed in the proximal tubule by this transporter called SGLT2. It's a sodium glucose co-transporter. So what that means is that, well, if you reabsorb glucose, you're not excreting it, it's going back into the bloodstream. So if you inhibit this transporter, you don't reabsorb that glucose, and that glucose is lost in the urine, and thus it lowers blood glucose, right? So in terms of anti-diabetes, it's pretty straightforward, right? It's a glucose lowering drug. The other thing that means, though, is because sodium and glucose reabsorption are coupled, it means that when you inhibit an SGLT2 transporter, you're also inhibiting the reabsorption of sodium. So I'll come back to that idea in a little bit. But this was generally what was understood about the mechanism. And yet when these drugs were first approved in 2013, this is from the FDA medical review of DAPA and canagliflozin, you can see that both of these drugs cause an acute drop in GFR. And then when you remove the drug, GFR tended to come back to normal. And so because of this acute drop in GFR, the labels initially prohibited the use of SGLT2 inhibitors in chronic kidney disease. And in addition, there was this kind of unexpected rise in hematocrit, and it was a dose-dependent rise in hematocrit that no one really understood. And so the questions were, what are the mechanisms? Why is this, SGLT2, as far as we know, is only expressed in the proximal tubule of the kidney. So why is this kind of simple mechanism leading to these changes in GFR, changes in hematocrit, and what are the long-term consequences of these kind of short-term changes? Interestingly, a couple of years later in 2015, the IMPA-REG trial read out, and this study was a cardiovascular outcomes trial that was just trying to show that empagliflozin, an SGLT2 inhibitor, didn't exacerbate or didn't increase cardiovascular events, right? But instead, it showed really quite an impressive reduction in the rate of GFR decline. So if you look, this is the placebo group, and this is the empagliflozin group. There's an initial drop, and then really a leveling off of GFR decline. And even more surprisingly, there was this really impressive reduction in heart failure hospitalizations, totally unexpected results at the time. And these results have now been repeated in multiple other trials with SGLT2 inhibitors. So Canvas in 2017, DAPA-HF in 2019, the Credence study, DAPA-CKD, IMPA preserved, IMPA kidney. So multiple trials now have shown that SGLT2 inhibitors reduce both rates of progression of kidney disease and reduce heart failure events. So while there was initially this concern, instead these outcomes trials have shown this large unexpected benefit in heart outcomes, and SGLT2 inhibitors are now becoming the standard of care for both HFrEF and chronic kidney disease, regardless of diabetic status, which I'll touch on. So we started working on this problem. So dapagliflozin was developed by AstraZeneca, and we started working on this problem back in 2014, 2015 with AstraZeneca. First, just trying to understand, before we had all these outcomes trials, what is the mechanism of this acute drop in GFR with SGLT2 inhibitors and this rise in hematocrit. And then ultimately in 2015, once there was the outcomes from the IMPA-REG trial, what's the mechanism for this reduced heart failure risk? So to do that, we had already been developing a model of cardiorenal function, and we applied, and through this process have expanded and much more developed this cardiorenal model. So I need to talk a little bit before I get specifically to this mechanism question for SGLT2 inhibitors. I need to talk a little bit about the model itself and how we developed it. Okay. So really, modeling of cardiorenal function just starts with translating what we know about physiology, representing it with fundamental equations, things like Ohm's law to describe pressure, relationships between pressure, flow, and resistance, Starling's equation, transport processes, mass balance equations, and many of the parameters for describing the system can be obtained from the literature. Other ones can be obtained from fitting. Just to give you an example of this, if we think about a really important relationship in the model is this relationship that describes filtration across the glomerular membrane, right? So for a single nephron, glomerular filtration rate is proportional to the ultrafiltration coefficient, which is a property of the glomerular membrane that's been measured. We know its normal value. The pressure in the hydrostatic pressure in the glomerulus, the oncotic pressure in the glomerulus, and then the hydrostatic and oncotic pressure in the Bowman space, right? So it's really a function of the pressure drop across this membrane. So that's just kind of an example of the types of kind of fundamental process equations that go into the model. All right. This is kind of a large overview of the model. I'll go through this super quickly. So first, we can represent the renal vasculature as a set of N nephrons in parallel. If we know something, if we know the blood pressure at the inlet and the pressure at the outlet, and we know something about the resistances of the preafferent and the afferent and the efferent and peritubular vascular arterioles, then we can calculate pressure at each point. And if we know the pressure inside the glomerulus, then this feeds into this model of nephron filtration and reabsorption, so we can calculate rates of filtration across the glomerulus. And then if we know something about rates of reabsorption of sodium and water and glucose and other electrolytes in each segment of the tubule, we can calculate rates of reabsorption and ultimately rates of excretion of sodium and water out of the kidney. And then that feeds into a larger model of cardiovascular function. So if we know water and sodium intake and water and sodium excretion, we can calculate the volume of blood, the concentration of sodium. We can use Starling equations to calculate the distribution of water and sodium between blood and interstitial fluid. And then if we know the blood volume, we can also calculate pressures and flow rates through the cardiovascular system. And we have two versions of the model. I would say this is the cardio light version. In this version, the heart is represented pretty simplistically. If I have time at the end, I'll mention briefly our kind of maybe capital cardio model, where we have a much more detailed representation of the heart. But for many purposes, the pretty simplistic representation of the heart that we have in this version is sufficient. And then again, the really big important part of this model too are all the regulatory feedback mechanisms that control different parameters in this model, right? So all the different regulatory mechanisms that control the resistance of the afferent and efferent arteriole, which are super important, for instance, in determining the pressure in the glomerulus, or factors that determine rates of sodium reabsorption and water transport in different parts of the nephron, for instance. Okay, and so how do we know, before we try to use this model to do something in drug development, how do we know that we have a good model, right? Well, first of all, I think about this as kind of this pyramid process, where first we need to describe the normal physiology of the system, the pathophysiological changes that occur in disease. Once we understand those things, then we can add on to that the pharmacologic effects of therapies of interest, and then we can use the model to do simulations. And this is not a thing where we ever really have a complete and final model. I think about this as a continuously evolving model, where as we continually integrate data into the model, we use that data to calibrate our model. We test our model with new data. Oftentimes we find some things fit, some things don't. So that leads us back to the calibration process. We find additional data, we incorporate it into the model, repeat, validation, things that don't fit leads us back to this calibration process. And over time, and we've been working on this model since, I've been working on it since 2009, and it really builds on a lot of work that was done prior to that at Entelos, and even prior to that by Arthur Guyton and many of the others who have worked in this field for a long time. So it's really a process of continual data integration. But there are certain checkpoints along the way that we have to meet to know whether we have a good model. And actually, anytime we make a change to the model, we have to go back and make sure that we're still meeting these criteria, right? So first is that it should be able to reproduce normal, stable baseline behavior. So normal values for cardiac output and mean arterial pressure and renal blood flow and GFR. So just making sure that those are all normally flat, stable, and in the normal range. One of the ways that we've calibrated many of the feedback mechanisms in the model is by testing the model's response to acute perturbation. So for instance, if you give an infusion of sodium into normal humans, there will be a change in sodium excretion, but that change in sodium excretion is really driven by many of the neurohormonal changes. So renin will go down, aldosterone will go down, vasopressin will go down. And so making sure that the model can capture these sorts of acute perturbations. This is over a few hours. We can do the same thing over a few days, right? If we give a salt load to subjects who are salt sensitive or salt resistant, can the model capture the difference in sodium excretion and blood pressure between those different individuals? Then we also want to see if we can, predict kind of, days to weeks responses, and particularly with many of the pharmacological interventions that we use to target these neurohormonal systems, right? So the RAS pathway is a big target of therapy. So renin inhibitors, ACE inhibitors, angiotensin receptor blockers, MR antagonists. So another test is if we can simulate the concentration response to these drugs, so changes in things like plasma renin activity, and then also the kind of integrated response to these drugs, which typically is changes in mean arterial pressure. So for these different classes of therapies, we have been able to show that our model can reproduce changes in plasma renin activity and changes in mean arterial pressure for different ARBs, ACE inhibitors, renin inhibitors alone and in combination. Okay, So from that point, we feel like we have a pretty good model of normal physiology. The next part is to start using the model to look at disease states. And through the course of this presentation, I'll go through these different kind of checkpoints for ensuring that we're reasonably modeling disease states. And then talk some too about how we incorporate new physiology into the model. Okay. But back to the SGLT2 inhibitor case study. So the disease here that we're interested in modeling is chronic kidney disease and heart failure. Today I'm going to focus much more on chronic kidney disease. There's a lot more to say about heart failure, but just to keep it a little focused, I'll be talking about chronic kidney disease. And we spent a lot of our, most of our development to date has been spent on understanding diabetic kidney disease, although we're also doing some work on IgA nephropathy and FSGF nephropathy and other kind of smaller types of kidney disease, or less prevalent types of kidney disease, I should say. But if you look at diabetic kidney disease, it's interesting because in the early stages, there's this hyperfiltration phase. And then the degree of early hyperfiltration, the more early hyperfiltration, the greater the subsequent decline in GFR. And then GFR declines progressively over time. And over time, albuminuria, or urinary albumin excretion, rises. And then later it falls just because GFR has fallen so low that you have very little filtrate to contain albumin in it. So if we want to model diabetic kidney disease, it's important that we understand the processes driving this. And actually, when we first started looking at SGLT2 inhibitors, we weren't even looking at SGLT2 inhibitors, we were just trying to understand the role of SGLT2 in this hyperfiltration phase. So Volker Valen back in 2012 published this tubular hypothesis of hyperfiltration. And the hypothesis is that when you filter more glucose, you reabsorb more glucose. And because you're reabsorbing more glucose, you also reabsorb more sodium and water. And that leads to changes in Bowman space pressure, so pressure in this capsule, and leads to changes in sodium flow through the macula densa, which then stimulates this tubular-glomerular feedback, causing afferent vasodilation, and that this afferent vasodilation is really what causes glomerular pressure and thus GFR to go up, right? So we just wanted to see, first of all, mathematically, could we reproduce this type of response in our model? Right? So what we did is we simulated a 15% increase in sodium reabsorption, because that's typically what you get when you, that's the amount of sodium that we think is reabsorbed through SGLT2 in the proximal tubule. And what we found is that when you give a 15% increase in sodium reabsorption, right, that's essentially like instantaneously giving diabetes, there's an increase in single nephron GFR, and that's in agreement with the hypothesis, right? And that's really driven by this, decrease in the TGF signal. But the interesting thing is that that initial increase is not super big. And the other interesting thing is that when we look at this acutely, it looks like we've reached steady state, but we're not really at steady state, right? So over the short term, this TGF response happens very quickly. But what we can see is that we're not at steady state because we're not in sodium balance. So because we're now reabsorbing more sodium through SGLT2, there's a sodium imbalance. And as I said earlier, the kidney is always going to return sodium balance. So then we thought about what's going to happen if we extend the simulation out over time. Well, what happens is if you have a nice healthy kidney that has a really strong ability to respond to those neurohormonal signals, then sodium balance is returned really quickly, and you don't really get any additional increase in single nephron GFR or in glomerular pressure. But if that natriuresis response, if that regulatory response is impaired, and we know that it's impaired in patients with diabetic kidney disease, then it takes longer to return sodium balance, and that sodium balance is returned at a higher pressure, right? So we think that really what's happening in early diabetes is you're increasing the amount of sodium that's being reabsorbed. The kidney is not able to respond to regulatory signals in a healthy way. So you end up retaining sodium and water until the glomerular pressure rises enough to filter enough sodium to return that sodium balance. That makes sense. Okay, so that was really interesting. If you think that the initial, and so, we know in diabetes, what we think happens, if I go back to the slide, right? you have this early hyperfiltration phase, and that hyperfiltration phase is an indication that you have glomerular hypertension, right? Pressure in your glomerulus is high, and we think that that's what causes faster nephron loss and a faster rate of GFR decline over time, right? So if that's the case, then it should be as simple as just inhibiting SGLT2 to kind of reset that sodium balance at a lower level and return homeostasis, right? So when we simulated this, we had some data from our collaborators at AstraZeneca in healthy subjects treated with an SGLT2 inhibitor, where they measured serum creatinine and GFR and sodium and water excretion. And what we found was that the direct inhibition of SGLT2 was not really sufficient to account for that rise in serum creatinine, which is an indicator of a fall in GFR that you see. And this is a little bit surprising because I thought this was going to be straightforward. But what happens is really you decrease the amount of glucose being reabsorbed, but you shift that reabsorption downstream to SGLT1. SGLT1 is another transporter that usually isn't doing very much, but once SGLT2 is blocked, then suddenly all of that extra glucose or extra sodium gets reabsorbed through SGLT1. So we don't think that that alone is the mechanism. So then that kind of led us to thinking about, well, what else do we know about SGLT2? Another thing we know about SGLT2 is that it's co-localized, and we've kind of grown to understand this a lot more over the the last decade. The SGLT2 actually interacts with NHE3, and NHE3 is a much more powerful sodium reabsorber in the proximal tubule. So we think what's really happening is that when you inhibit SGLT2, you also indirectly block NHE3, and that was able to partially account for the response. The other thing that happens is that you end up with glucose remaining in the tubule, and that glucose that remains in the tubule is normally not there, right? Normally you reabsorb it all in the proximal tubule, but when it remains and flows through the tubule, well, glucose is osmotically active, so you end up with glucose showing up later in the nephron, and that causes this osmotic diuresis effect that leads to additional water excretion. And then additionally, this kind of interesting thing happens where you get an increase in water excretion without very much change in plasma sodium concentration. And in order to account for that, we have to assume that there's some shifting of sodium into this peripheral storage compartment. And this is another thing that we've grown to understand a lot more over the last decade, is the way in which sodium can be shifted out of the blood and interstitium and stored interstitial stored non-osmotically and then released over time. And so when we incorporate these 4 different components of the model, we're able to reproduce kind of the totality of the data that we have. And what was really interesting was that, so if you think about this, is a little bit of a confusing figure, but what we're looking at here, if we look particularly at glomerular pressure, which glomerular pressure is the big driver of kidney disease progression, right? And then interstitial fluid volume. If you think about heart failure, one of the main causes of heart failure hospitalization is that patients have excess interstitial fluid volume, they're congested, they get so congested that they can't breathe, and they show up at the hospital, right? So when we included all 4 mechanisms in the model necessary to explain the data, what we found, and that's the green curve here. So the red line is if we just had direct inhibition, the blue is if we added in NHE3 coupling, the gray is if we also account for osmotic diuresis, and then the green is the shifting of peripheral sodium storage. And so you can see that when we incorporate all 4 of those mechanisms, we first of all predict that SGLT2 inhibitors are going to drop glomerular capillary pressure. And interestingly, and the thing that we didn't really expect, but was really kind of in agreement with those unexpected reduced heart failure hospitalization data, was that with this mechanism in place, the model predicts that SGLT2 inhibitors are going to really reduce interstitial fluid volume. And interestingly, it predicts that they're going to do that a lot better than other types of diuretics. So bumetanide is a typical loop diuretic that is used to treat heart failure, and it decreases interstitial fluid volume quite well. But the problem with it is that it increases it at nearly the same, or it decreases it at nearly the same ratio that it does blood volume. And in heart failure, that's kind of a problem because you want to reduce congestion, But if you lower blood volume too much, you lower filling of the heart, and you can actually reduce organ perfusion. So the model predicted that SGLT2 inhibitors do a better job at decreasing interstitial fluid or congestion without getting excess reduction of blood volume. And that's part of the reason that we think that it's so successful in reducing heart failure hospitalizations. Okay, so all kind of coming back to our major challenges, right? We were able to look at this kind of biomarker, this change in GFR that was unexpected. So that initial drop in I think I didn't quite connect that point very well, but that initial drop in GFR that we see is actually a consequence of dropping the glomerular pressure, right? And so when you drop that glomerular pressure, you reduce the rate of glomerular injury over time, right? And so the model was able to explain that we get that initial drop in glomerular pressure in GFR because we're dropping glomerular pressure, not because we're doing something detrimental to the kidney. And we're actually able to link that concerning biomarker to functional improvements. And this provided a kind of a physiological basis for why SGLT2 inhibitors are improving these outcomes. And it actually relieved a lot of the safety concern over that drop in GFR. But it did then raise new questions, okay? So this acute drop in GFR is because we're dropping glomerular pressure, and that's fine in patients who have normal kidney function. But what about in patients who already have a really low GFR? You know, if you have a low GFR and you're getting a 5 to 10 milliliter drop per minute drop in GFR with this drug, is that going to be problematic? And also in patients with low GFR, they actually filter less glucose, and they actually have, it's established that they excrete less glucose with SGLT2 inhibitors. So there was some concern that the efficacy may be diminished in patients with low GFR. And then there were also questions about whether SGLT2 inhibitors would be beneficial in non-diabetics with chronic kidney disease. All right, so we wanted to be able to understand some of these things, not in healthy subjects, but in patients with chronic kidney disease. So I need to spend another minute or two talking about how we model those processes of diabetic kidney disease, right? So if we think about what we know about diabetic kidney disease, there's glomerular hypertrophy, there's thickening of the basement membrane, there's glomerulosclerosis. Over time, there's podocyte injury and loss, and then there's injury to the proximal tubule and development of interstitial fibrosis. So if we want to relate this signal of diabetes to kind of these changes in glomerular and renal tissue that we see over time, well, we need to think about really what's causing GFR to decline functionally, right? So if this is our equation for GFR. We know that in diabetic kidney disease, so it's a function of how many nephrons you have, how permeable that glomerular membrane is, and how much surface area it has, that's KF, and then the net filtration pressure. Well, we know that in diabetes, glomerular pressure is actually going up, not down. And we know that when there's podocyte and protein filtration, if anything, that's going to cause the Bowman space pressure to go up. So this net filtration pressure is going up, right? So in order for GFR to go down, there are really only two ways, right? You can either lose nephrons or have a decrease in KF, or some combination of both, right? So what we really think is happening is that there are these kind of two processes, right? There's some changes in the surface area and the permeability of the glomerular membrane that leads to changes in KF. And those can themselves contribute to loss of glomeruli, which can cause you to lose a nephron. Or there can be processes like tubular atrophy and fibrosis that cause tubule loss, right? So we then needed to connect diabetes, this process of diabetes, to this process of glomerular injury and nephron loss, right? Well, we know in diabetes that there's an increase in pressure in the glomerulus as a consequence of that compensation for increased reabsorption. And we know also that because there's hyperfiltration, right, each nephron is filtering more, that nephron is having, that proximal tubule is having to do a lot more reabsorptive work than it normally does. And that reabsorptive process is active. It requires a lot of ATP. And so in a kidney that's hyperfiltering, you're really overworking that proximal tubule, and it's using up more ATP than it has fuel to generate. So there's this ATP deficiency. And this glomerular hypertension process initially causes glomerular hypertrophy, but over time causes segmental glomerulosclerosis. This excess proximal tubular work can cause tubular inflammation and atrophy and fibrosis. In addition, high pressures in the glomerulus cause podocyte injury. Podocytes are the cells that normally keep protein from being filtered across the glomerulus. But when you injure those podocytes, first of all, that podocyte injury itself can cause sclerosis of that glomerular membrane. And podocyte injury also results in leakage of protein across the glomerular membrane. You don't normally have much protein in your tubules. So when you end up with a lot of protein in the tubules, that stimulates tubular inflammation, atrophy, and fibrosis. So all of these processes, we can essentially define rate constants that describe the rate of progression of each of these processes in response to each of these signals. And what happens then also, though, is as you lose nephrons, there's also this kind of cyclical process where when you lose nephrons, the kidney's still trying to maintain the same amount of sodium and water excretion. So what happens is when you lose nephrons, the remaining nephrons compensate by filtering more. They do that at a higher pressure. That higher pressure leads to compensatory hyperfiltration, which leads to even further additional proximal tubular work. And so it's kind of the cyclical process that once you start, you may slowly lose nephrons over time, but it kind of accelerates as you go. And so if we give in our model this stimulus of diabetes at time 0, we can show that with these relationships, we can reproduce the natural history of diabetic kidney disease. So essentially, you give diabetes at time 0, you see hyperfiltration, so GFR goes up, and then progression of GFR over time. There's this increase in proximal tubular work, and it gets worse over time. Early on, there's very little proteinuria, but as the pressures in the glomerulus rise, it starts damaging the podocytes and driving up proteinuria. And also early on, there's this hypertrophy, so the ultrafiltration coefficient goes up, but then as you have glomerulosclerosis, it goes down. So it kind of captures all of these processes that we know about what diabetic kidney disease looks like. And if you think about this, this is one trajectory, but if we change the severity of diabetes, we can get many different trajectories. Or if we sample at different points along this trajectory, we can get patients with different kind of existing levels of injury. And so you can think about each of those trajectories and each of those points as a virtual patient. Okay. So if we make a population of virtual patients, we first need to make sure that those virtual patients, before we try to look at what SGLT2 inhibitors do, we need to make sure that those virtual patients behave like we expect. So we spent a good bit of time in the past looking at some of the seminal trials in diabetic kidney disease, so rationale and IDNT, and making sure, first of all, that we can represent the baseline characteristics of those studies, and then that we can represent the changes that we see in response to these kind of standard of care treatments in albuminuria and GFR decline over time. So then once we feel like we can represent the baseline distributions and the short-term response, we also need to make sure that we can, then we can start to use the model to look at SGLT2 inhibitors. So one of the things that we did was that there was a trial, it's been completed now, but the DAPA-CKD trial was being planned. This trial had 3 arms, 2 in type 2 diabetics, one group with normal kidney function and one with impaired kidney function, and then one group of non-diabetics with impaired kidney function. And so the questions that we wanted to ask were, if you look at these patients with impaired kidney function, is this acute GFR, drop in GFR going to be problematic? And what we could show was that when you give an SGLT2 inhibitor in normal versus impaired kidney function, the GFR drop is actually going to be a lot less, but the reduction in glomerular pressure is going to be sustained. So you're still going to get a reduction in glomerular pressure, but that's not going to translate into the same drop in GFR. So that gave a lot of confidence in the ability to go into patients with impaired GFR, expecting to have beneficial effects on kidney disease without that acute response detrimentally affecting their kidney function. In addition, we know if you look at the urinary glucose excretion, it's well established that it's reduced in those with low kidney disease at low kidney function. So there was some concern that the efficacy would be diminished, but again, we still see a substantial drop in glomerular pressure. And if we look at the reduction in interstitial fluid volume, that's sustained as well. And then lastly, a big question was whether SGLT2 inhibitors were going to be efficacious in non-diabetics, because all trials up until this point had been in only diabetic patients. And our simulations suggest that non-diabetics were still going to see substantial reduction in both interstitial fluid volume and in their glomerular pressure and in their albuminuria reduction. So this provided a lot of confidence for proceeding to phase 3 trials. And actually the AstraZeneca trials in HFrEF and CKD were the first trials that weren't limited to diabetics. So the DAPA-CKD and the DAPA-HF trials included both diabetics and non-diabetics. I think I'm running a little bit out of time, so I'm just going to skip quickly through these. This is just some of the calibration we did to ensure that the model is progressing in agreement with the available data. We were then able to match our virtual patients to the population of the DAPA-CKD trial. And then one of the coolest results that we obtained was, and this is maybe my most exciting slide that I've ever produced, is we were able to reproduce in both the placebo group and in the SGLT2 inhibitor group the rate of change in GFR over the long term. So this is a simulation, a virtual population simulation of the DAPA-CKD phase 3 clinical trial with dapagliflozin. And so first of all, this shows that we have really an understanding of that rate of decline of GFR. And this is really important now because a lot of the FDA is now accepting, beginning to accept changes in GFR slope as an endpoint, as opposed to the hard endpoints of the past, like doubling of serum creatinine and end-stage renal disease. And so we can reproduce that slope in placebo patients and also the dynamics of that change in GFR with SGLT2 inhibitors. And also we were able to simulate events of this trial, so doubling of serum creatinine and or 50% eGFR decline. And our simulated hazard ratios were very much in agreement with the actual outcomes of the DAPA-CKD study. And interestingly, the DAPA-CKD study confirmed that both diabetics and non-diabetics benefit from this drug. One thing we still don't understand is actually the diabetics in some cases seem to respond even a little bit better than the diabetics. I have some hypotheses about why that's the case. I think it has something to do with the level of severity of patients in these trials, but nevertheless, I think it was kind of a cool thing that we were able to show prior to the trials that we would expect non-diabetics to have efficacy with this drug. Okay, so we were able to reproduce the long-term progression of the disease and reproduce the rates of outcome and risk reduction in these seminal clinical trials. And importantly, I think when we think about de-risking these expensive trials, I think the modeling provided mechanistic support and rationale for the expanded indication, including non-diabetics, CKD in this phase 3 population. Some of the work that we're doing now is we're continuing to use this model. We're working on looking at SGLT2 inhibitors in combination with many of the new drugs in development. So we're doing a lot of work now with endothelin antagonists, with relaxin and other mechanisms of action currently in development. I think I'm out of time for I want to leave a little bit of time for questions. So I might skip through the last of these slides, but this is just kind of an overview of the application of this model to support dapagliflozin. Oh, I did want to quickly mention that we also use this model, after the DAPA-HF study and the DAPA-CKD study, particular health authorities, so for heart failure, the Japanese health authority wanted some additional data on the higher dose of dapagliflozin. So they were concerned that the higher dose wasn't needed. And we were able to do some simulations of interstitial fluid volume that were able to convince them to not require an additional study looking at the dose response. And similarly with DAPA-CKD, one of the challenges of this study was in the Chinese subpopulation. For kind of some complex reasons, there was a fairly short amount of follow-up time. So the average follow-up time in the Chinese subpopulation was only 3 months. So there was some concern about whether these results were really applicable to Chinese subjects, but we were able to match our virtual patients to the Chinese subpopulation and show that they were expected to follow the same trajectory and see the same benefit as the general population. Because you can think about it, if you only have 3 months, right, you're still in this. If you looked at just the Chinese subpopulation, they just had a decrease in GFR, right? And we were able to show that if you followed it over time, they should follow the same rate of trajectory. And so that resulted in them accepting dapagliflozin without requiring a longer additional follow-up study. All right, I think I'll leave it there. There's only 5 minutes left. So thank you guys for your time, and at this point, I'll take any questions.
Thank you, much appreciated. We do have some questions from the audience. One of these is a technical modeling question, which is, this is appears to be a multi-scale model, which is a stiff system. And does it take a long time to solve simulation in the platform, given the different timescales you're trying to reconcile?
Yeah, so that's a great question. So we have 2 different versions of this model, and we have it for a reason. So we have like the cardio light version, and then in this model, everything is really on the order of minutes or longer, and it actually solves really fast. So I can simulate, a year of a virtual patient in, about a second in this model. But we also have another version of the model that is has a full simulation of the cardiac cycle. And that introduces, so first of all, there are just some timescale issues, right? That the cardiac cycle happens, there's 1 beat every second. And also there's no steady state, right? It's always cycling. So this model is very stiff, and it takes not an excessive amount of time to run, but I would say it takes about 3 minutes to run probably 3 months of one virtual patient. So a good bit longer than this one.
Okay. Thank you. Another question is, would this model be able to simulate the outcome from natriuretic peptide synthesis modulation? So in other words, combination therapies with other drugs that are intended to modulate, blood pressure.
Yeah, so I would say that, yes. So we've looked in the past some at, if we're talking about natriuretic peptides, we looked some at Entresto. So we have a representation of A and P in the model. For other mechanisms of action, I would say if any drug whose primary action is in some way altering either directly or indirectly altering resistances and reabsorption rates, things like that, I think it's fairly, yes, the model can easily be used to represent those. I think for other sorts of drugs, so for instance, we're thinking now a lot about IgA nephropathy, and so in IgA nephropathy, a lot of the drugs that are being developed are more, targeting particular antibodies, where it's a little bit more difficult to get at that. But for anything that has primarily some form of hemodynamic mechanism, I think the model is really well suited for looking at those types of combinations. For other types of indications, it's not that we can't do it, but we have to think a lot more about how those markers ultimately, in some way, impact this process, right?
Great. Thank you. And just a comment for the audience that the slides will be available on the ROSA webinar site if you wanted to look at some of these questions in more detail. We have another question from the audience. Do you think that this platform could incorporate molecular level findings from the standpoint of patient variability and personalized medicine? And if so, how would you do that, either implicitly or explicitly?
I think to some extent, yes. So I think one of the things that we're really interested in is trying to understand, I mean, even the slide I'm on, right? If you look at the gray lines here are data from real patients, right? And there's clearly a lot of variability in their rate of eGFR decline. And our model can represent that variability by representing changes in different rates of different parameters, but often we don't know the underlying molecular factors that cause that variability, right? We just know that those parameters vary, right? So for instance, proximal tibial sodium reabsorption, the rate can vary from 40 to 80% from individual to individual, and it's likely because of big differences in their rates of transporter expression in just as an example. Another example is that FSGS nephropathy is related to the ApoE mutation, and we don't really understand why, but we're beginning to understand that molecular implications of that have impacts for these, essentially for what these rate constants are. So I think it's not something that we've done much of yet, but I think it's something that I certainly have a lot of interest in thinking about in the future, how we can really understand more about why individuals both progress so differently and also respond differently to therapeutic interventions.
Great, thank you. Well, I think that's all the time we have for questions. I'll remind the audience to please check the ROSA website for upcoming webinars. We'll try to get to questions, other questions through email. And I want to thank everybody for joining, and I want to thank Melissa for a wonderful presentation. And take care, everyone, and have a nice day.
Thank you, guys.
Thanks, everybody.